students, hope you all are doing well and you are understanding whatever I am teaching you. So today we are going to deal with a very interesting chapter, which is genetics. Now, what is genetics? From the word itself, you can understand that it deals with something about genetics. Yes. So, genetics is the study of transmission of body feature from parents to offspring, which is ultimately the study of genes. Why do we need to study about genetics? Now, genetics deals with a few things like genetic engineering and genetic counseling, which contributes a lot in today's modern science. Now, what is genetic engineering? Genetic engineering deals with the alterations in gene artificially so that we can get new products, which helps in different purposes. Now, what is genetic counseling? Genetic counseling deals with the early counseling of different diseases so that it can be prevented even before the birth of a child. Now, before reading about genetics, we need to know about two things, that is character and traits. Now, what is character and traits? Character is something which character is the features that we inherit from the parents, and traits are the alternative forms. For example, the color of an eye can be brown or blue, but now you might have noticed that brown is, for example, the color of an eye may be brown or blue. Now you might have noticed that the brown is the most inherited eye color, while blue is a very rare one. So here we can understand that brown is the character here and blue is the trait. Likewise, the color of a vision, which is normal, is a character, and color blindness, that is red or green color blindness, is also the trait of this character. Now, let us first move on to chromosomes, because as we all know that chromosomes are the carriers of the genes. Now, there are two types of chromosomes, but first we need to know that But for now, first let us start with the chromosomes, because we already know that the chromosomes are the carriers of the genes. Now, those homologous pairs are of two types, which are autosomes or autosomes chromosomes and the sex chromosomes. So what are the autosomes? Autosomes are the identical chromosomes which does not differ in any way and they stay in pairs. Now, what are those sex chromosomes? Sex chromosomes are a bit different from each other, like the pairs are a bit different from each other. It exists in two types, that is the X and X type and the X and Y type. The X and the X type is the female chromosome and the X and the Y type is the male chromosome. Now we can understand why it is known as the sex chromosome, since it's determine the sex of a offspring or the parent. Now, how does this determination of the sex chromosome occurs? Like how is it carried from the parents to the offspring? Now let us understand that. So as here we can see that the female or the chromosome of a mother is the XX type. You can see that the mother has XX type chromosome and the father has XY type chromosome, and we know that one member of each of the pair comes from the homologous chromosome and joins to form a chromosome for the offspring. Now, as we all know that the sperm travels to the ovum and the fertilization occurs and the offspring is produced. So if the X chromosome from the father fertilizes the mother ovum, it becomes a female, which is X joined with the X chromosome. While if the Y chromosome from the father joins with the X chromosome of the mother, which is XY, it becomes the male. Now, to understand the concept of the sex determination of the offspring from the parents, I hope you did. Now, after discussing about the chromosomes, let us move on to the alleles. Now, what are alleles? Alleles are nothing but the alternative gene forms occupying the same position on the homologous chromosomes, affecting the same characters but in a different or alternative way. Okay, now alleles are of two types, which are dominant and of recessive. Now why is it known as dominant allele and the recessive allele? From the name itself, you can understand that the dominant allele can easily express itself, but there is a condition to this recessive allele. What happens when two recessive alleles stays together? It can easily express itself. There is no problem. But if the recessive allele stays with the dominant allele, then it cannot express itself. So, dominant allele is expressed in the capital letters, like you can see here, and the recessive allele is expressed with small letters, as you can see here. Now, here you can see three types of things. What are those? Two capital A's, which represent two alleles, but both are dominant here. Since you can see here, both are represented by the capitals. So, it is known as homozygous. Homozygous term means similar. It is known as homozygous dominant. Here you can see there are two small a's, which means both are recessive. So, it is known as homozygous recessive. But here, as you can see, what I was talking about earlier, here there is a dominant allele and recessive allele. And as you know, if both are present, then only the dominant one can express itself. So it is known as heterozygous. Heterozygous means dissimilar. It is known as heterozygous dominant, since the recessive character cannot express itself. But remember that the chromosome as a whole can neither be dominant or recessive. Okay. Now, here you can see a term called genome. Now, what is a genome? Genome is nothing but the full complement of DNA. It means it includes all the genetic and intergenetic regions. Okay. Please remember this one. It is very important. Now, we need to know about the two things, which are the genotype and the phenotype. Now, what is genotype? Genotype is nothing but the genetic constitution, that is the actual set of genes present in the chromosome. It doesn't matter if it is recessive or dominant. All of them are included in this genotype. But what is phenotype? Phenotype are the only expressed character which can be observed. That is, we know that if there are dominant and recessive characters, only the dominant one is expressed. So the phenotype will only deal with that. Now let us understand this concept more clearly with some example. In a square formed box, so that we can easily determine the genotype and the phenotype. Okay, now let's make it more clear with this example. Now, suppose many of us can roll our tongues, but many of us cannot. How does it happen? And how is it inherited from our parents? Let's discuss this. Now, suppose there's a mother who have the heterozygous dominant gene and the father who have the homozygous dominant gene, which is denoted by the capital R and small r, and both of them are denoted by capital R's. Now, here we make the Punnett square and write it like this. That is both the capital R's for the father and the gene of the mother. Now, how does the Punnett square happen? It's nothing but we have to multiply this one with this one, and again we have to multiply with this one with this one and write it here. Just like simple multiplication. Okay. Likewise, we have to multiply this one with this one and write here, and this one with this one and write here. So, how does it become? So, here we get one homozygous dominant, another homozygous dominant. Here we get heterozygous dominant, and here we get heterozygous dominant. Okay. Now, obviously, the genotypes is 2:2. Why, you may ask? It's simple, because there are only two types, which are homozygous dominant and heterozygous dominant, which are the only genetic constitution. That is why the genotype is 2:2. Nothing complicated about it. But the phenotype here is 4:0. Why? Because here you can see although there are recessive traits, but you know that recessive traits cannot express itself. So there are only two types, only one type, which is dominant. That is, all of the offspring of these two parents can roll their tongue. So the phenotype becomes 4:0. Now let us take another example where the mother has the heterozygous dominant and also the father has the heterozygous dominant. We arrange the Punnett square in the similar way and we get one homozygous dominant, two heterozygous dominant and one homozygous recessive. So the genotype becomes 1:2:1. 1 for the homozygous dominant, 2 for the heterozygous dominant and 1 for the homozygous recessive. Since genotype is the genetic constituents, that is all of the characters can be represented by the genotype. But since in phenotype it is the only observable characters, the phenotype becomes 3:1 because all three of them are dominant. Only one is recessive. So the phenotype becomes 3:1. So we can say there are 75% chances that the offspring produced by this parents can roll their tongue, while 25% of them cannot roll their tongue. Now, because and you need to know one more thing, that for tongue rolling, the who can roll their tongue are the dominant ones and who cannot roll their tongue are the recessive ones. Okay, got it? I'll also attach few other examples with the notes. And notice that all these two examples are something which are dealing with the autosomes. Okay, and inheritance. Now, what is sex-linked inheritance? Sex-linked inheritance is nothing but it occurs due to the X and the Y chromosome, which are the sex chromosomes. Now, there are few defects or diseases occur due to the presence of some recessive genes on the X chromosomes, and those are denoted by X0. Let us understand that concept with few examples. Now let us take a case of color blindness, and we have plotted three cases, each of different types. Okay. Now before going to that, you have to understand that if there is one recessive chromosome for the mother, then the mother is a carrier of that disease. But if both of the genes are recessive, chromosomes are recessive, then the mother is also affected with that disease. But for the father, since it has only one X chromosome, if it has one recessive chromosome, then the father itself will be the affected one. Okay. Now from here we can see that we have a healthy mother which is denoted by two X chromosomes, and we have a affected, that is a colorblind father denoted by X0 and Y. So let us plot it on the Punnett square and see what happens to the probability of the offspring being colorblind and not being colorblind. Okay. So here we write as X0 and Y, and here we write X and X. This is the female and this is the male. Okay. Now let us fill the Punnett square. So for the first box it becomes X0, X, this with this, and after multiplying this X0 with this one, it also becomes X0, X. Similarly, X0, Y. Similarly, the Y from here multiplies with the X to form X and Y, and here the Y multiplies with the X to form X, Y. Now, here you can see, as you can see here, we have two probabilities of daughter and two probabilities of a son. Here, both the daughters have probability of being the carriers of color blindness, and both the sons will be healthy. Okay. Now let us move on to the second case where we have a carrier colorblind mother and a healthy father. So let us jot down the points. Here we will write as X, Y and X, X naught. So what happens here is multiply with x, then we got here x with x naught, we get here x and x and x and y, we get x naught and y. Now as you can see here, here is also probabilities of two daughters and two sons. Okay, so the probability of the female being colorblind is almost equal to 0, because here we have only one recessive gene. That means it will be only the carrier of colorblind and not colorblind herself. That's why the daughters cannot be colorblind. But here you can see we get a healthy son but an colorblind son also. So the chances of the son of these parents being colorblind is 50-50, since one of them is healthy and another one is colorblind. Okay. Now for the third case here, we get a carrier of colorblindness mother and the affected father or the colorblind father. What happens here? Let's see. X naught y, x naught x. So x naught, here we get x naught x. Now here we get x naught y, and here we get x and y. Okay. So the probabilities of the daughter being colorblind here is 50, since one of them is a carrier of colorblindness and one of the daughter is colorblind, because both of the X chromosome have the recessive trait. Similarly, for the sons, the chances are also 50-50, since we have a healthy son here and because we don't have any recessive trait, and here since the X chromosome carries the recessive trait, that son can be colorblind. Okay, now did you understand the concept? John Mendel is known as the father of genetics, obviously, because he contributed a lot in the genetical field. Now there is a thing called Mendels Experiment, where he took some specific characters of pea plant, like the tall plant and the dwarf plant, or the wrinkled pods and the round pods, the green pods and the yellow pods, and he breeded those to find out the possible outcomes. For that, we need to know about two things, that is the monohybrid cross and another thing is the dihybrid cross. Now first, let us discuss about the monohybrid cross, where he has taken only one character in. Okay, so here he is taking a pure tall pea plant, which is denoted by the dominant genes chromosomes, and a dwarf plant, which is denoted by two small t's. And here you can see both are pure root. So if we interbreed them or cross them, we get a hybrid tall plant, which is denoted by a capital T and a small t. Or we can also call it a heterozygous tall plant. This is known as the F1 generation. Okay. Now, as we get this hybrid tall, let us interbreed it. Okay. That is, let us interbreed the heterozygous tall plant with another heterozygous tall character. So let us form the Punnett square here. Here we get the capital T and the small t, capital T and the small t. After the Punnett square is formed, we get one homozygous tall plant, two heterozygous tall plant, and one homozygous dwarf plant. This is also known as the F2 generation. Okay. So what happens to the phenotype? The phenotype becomes 3:1 because 3 of them are tall and 1 is recessive, and the genotype becomes 1:2:1 because there are three types of genes, obviously. That's why it becomes 1:2:1. Now monohybrid cross is comparatively easier than dihybrid cross, but don't worry, dihybrid cross will also be taught you. Okay. So now let us discuss about the dihybrid cross. Now what happens in dihybrid cross, that Mendel had taken two characters and crossed them so that we can get some different result. Now he has taken two characters which are the shape of the pod and the color of the pod of the pea plant. The dominant ones are the round and yellow, and the recessive ones are the wrinkled and green. Okay. Now before moving on to with the dihybrid cross, notice one thing, that all the characters are represented by the same letter, although the one is capital and one is the small letters, but both are the same letters, so that it doesn't become confusing. Okay. So let us take a pure round yellow and a pure wrinkled green. What happens in the first generation is that we get a heterozygous round and heterozygous yellow pea pod. We get four types of these. Those are R, capital Y, small r, small y, small r, capital Y, and capital R, small y. Okay. Now let us draw the Punnett square and let's see what happens to them. For the first one, we get capital R, capital Y, capital Y, which is a homozygous yellow and round pod. For the next one, we get capital R, small r, capital Y, small y, which is a heterozygous round yellow. Next we get R, Y. Similarly, we will complete the Punnett square. Now we have completed the Punnett square in this way. For this, the phenotype becomes 9:3:3:1. But the genotype is much more complex and you don't have to know about it right now. That's for the higher class, okay? So after jotting down those monohybrid cross and the dihybrid cross, Mendel found three laws related to the inheritance. Those are law of dominance, law of segregation, and law of independent assortment. Okay. So what happens in the law of dominance is that when a pair of character is present, that is one with the dominant allele and one with the recessive allele, only one can express itself. That is, we have already known, that is the dominant one, and the recessive one remains suppressed. Now what happens in the law of segregation is that a pair which has two members separate during the formation of a gamete, as we have seen both in the F1 generation of the monohybrid cross and the dihybrid cross. That is, the dominant allele and the recessive allele separate during the formation of a gamete before the F2 generation. Got it? Now, what happens in the law of independent assortment is that if two types of pairs are present, or rather two characters are present, one character will not interfere with the other character. Okay, that is when we saw that we had a round yellow pea pod and a wrinkled green pea pod. The round or the wrinkled allele will not interfere with the yellow and the green allele. Got it? What was the outcome of Mendels experiment? We got to know about different progenies and different variations, and we could also create some hybrid varieties of the pea plant, and not only that pea plant, we could create the hybrid varieties of many objects. Got it? Now let us get familiar with the term called mutation. Now what is mutation? Mutation is nothing but the sudden change in the set of genes or the number of chromosomes due to various effects. Now sickle cell anemia is a type of mutation, and why? We'll read that in the higher class. Okay. I will also attach some examples of mutations in the given notes. Okay, got it? Now with this, we end this chapter. I hope you have clearly understood the concept.